A very good morning to all of you. I am Dr. Prithviraj, HS, Assistant Professor of Botany, Sri Mahadeshwara Government First Aid College, Kollegala. Today I will be talking on a topic called Basics of Plant Tissue Culture. This has been included in the botany syllabus of final year students. Well, plant tissue culture is nothing but the in vitro culture technique. In this technique, plants can be grown inside the glass vessels like test tubes or conical flasks or any other glasswares that support the growth of the plant. Well, this technique was for the first time determined by Gottlieb Haberland. Well, plant tissue culture technique is dependent primarily on three important concepts called polarity, plasticity, and totipotency. Well, plasticity is the ability of a cell to convert from one cell to another cell, whereas the totipotency is the ability of a cell to develop into a whole organism. Well, to express totipotency, there are two important processes that occur during totipotency. One is the dedifferentiation and another one is redifferentiation. So dedifferentiation is the conversion of mature cell into a meristematic state and forming a callus, and again this callus can be redifferentiated into a whole plant or a plant organ. Well, explant is any part of the plant that is used for the culture. It can be a leaf part, it can be a stem part, or it can be any part like the flower or a flower, anther of a flower or ovary, etc. So this goes for dedifferentiation to result in the callus, which is nothing but an unorganized mass of cells, and this callus will become the entire plant through the process called redifferentiation. This technique requires growth regulators. So growth regulators are nothing but the hormones, and they influence the growth and development of a plant cell in vitro. They are the organic compounds that can be synthesized, that are synthesized in fact in the plants naturally, or they can be synthesized artificially also. So here there are some examples of some growth regulators: auxins and cytokinins. Auxins are AA, NAA, IBA, and 2,4-D, and cytokinins are BA, kinetin, 2IP, zeatin, and there are other hormones like gibberellins and ethylens that can also be used in tissue culture for the growth. Well, another important hurdle in plant tissue culture is the condition where the plants are grown or where the transfer of explant is carried out. So the place where the transfer of the explant is done onto the media should be free from any contaminants, particularly the microorganisms. Therefore, sterilization is a technique that is employed in almost all the tissue culture laboratories for the successful growth of the plant. So sterilization is nothing but it's a treatment that frees an object or an explant from the microorganisms, right? So it can be done either by the physical methods or by the chemical methods. Physically, sterilization can be done by using the filtration techniques or heat sterilization techniques. Chemically, it can be done by using some chemicals like sodium hypochlorite, mercury chloride, bromine water, etc. Well, the explant that is selected for tissue culture should be sterilized as that of any other material that we use for the tissue culture. So it is called as surface sterilization of the explant. Here the explant that is selected is treated with some surface sterilants like sodium hypochlorite, calcium hypochlorite, hydrogen peroxide and bromine water. So they are started with a piece of whole plant. So the piece of plant that is generally brought from the field is contaminated with microorganisms. So therefore, this is an important step to eliminate the contaminants from the explants. So this, as I told in the beginning itself, tissue culture requires a medium as a growth medium, right? So medium is nothing but a supportive area for the growth of the explant, and that includes inorganic nutrients, carbon source, growth regulators, organic supplements, vitamins, amino acids and gelling agents. It can be a liquid medium or it can be a solid medium. So the difference between the liquid and a solid medium is, in solid medium generally agar is used to solidify the medium. If agar is deleted, the medium will remain as a liquid medium, and if that is the case, then instead of explant we'll use single cells for the growth of the plant, and that liquid medium has to be agitated continuously for aeration. So there are number of culture mediums that are used for tissue culture. The best medium that is suitable for most of the plants is Murashige and Skook's medium. There are other mediums like Gambark's medium, Niche medium, Lloyd and McGowan's medium, and WPM or woody plant medium. So what is callus culture? Let us take another important concept of tissue culture called callus culture. So callus is an amorphous tissue, or it is an unorganized mass of cells. So they are thin-walled parenchyma cells. So they can be redifferentiated into a whole plant, which I have already discussed in the previous slides. So by supplying the auxins exogenously, callus can be can be made into a plant either by somatic embryogenesis or by somoclonal variations or through suspension cultures. Here are some photos showing how the callus is initiated. So this is an explant which is called a stem explant, and you can see on either side of the explant you can see the growth of the callus, and this is how the explant responds to the cultural conditions when it is provided with a medium, when it is sterilized properly, when it is treated with some chemicals, and when they are provided with the right laboratory conditions. This is how the plant will respond to the conditions. And this is a leaf callus. Leaf can also be used as an explant, and you can see the entire leaf has been covered by the callus, and you just cannot locate the leaf because that is how the growth of the callus occurs. It will completely change the structure of the leaf, and here you cannot make out which explant has been used. Well, this is the callus from the leaf explant. Another important concept that I would be emphasizing here is organogenesis. Generally, this is also an important concept, and it has been asked in many of the previous question papers. So, organogenesis is the formation of individual organs such as the shoots and the roots. So, the growth of the shoot is called as collagenesis, and the growth of the root is called as rhizogenesis. So, collagenesis is a type of organogenesis, and here you will find shoot buds that are initiated from the callus tissue. Okay, and in the rhizogenesis, the roots are formed from the callus tissue. If shoots are formed, it is transferred to the rooting medium, and if the roots are first formed, then it is very difficult to induce shoots. Therefore, in organogenetic process, we prefer the initiation of the shoots rather than the roots, because once the shoots are obtained, it is very difficult to root them. But when the roots are obtained in the beginning, it is very difficult to make them into shoots. So this is how the shoot regeneration happens in the in vitro conditions. You can see how these shoots have developed from the callus. So there are a number of shoots that has arisen from the callus. Here you can see the rhizogenesis or the root initiation from the explant. Generally, after the root initiation, the chances of getting shoots is very meager. So therefore, we generally discard those explants which produce roots initially. And again, you can see the callus and the roots that are produced. And here you can see the shoot regeneration from the nodal explant. And another important concept that a student has to know in tissue culture is somatic embryogenesis. Well, somatic embryogenesis is different from the zygotic embryogenesis. Embryo-like structures are formed from the somatic cells in the in vitro conditions. This is what we refer as somatic embryogenesis. So somatic embryos are larger in size than the zygotic embryos, and the plant growth regulator play an important role in somatic embryogenesis. It's of two types: direct embryogenesis and indirect embryogenesis. In direct embryogenesis, the callus are predetermined to become the embryos, and particularly the nucellus, the hypocotyl, and they are already predetermined for embryogenesis, and they can be induced to become embryos. And some other type of cells has to be subjected for embryogenesis, and it will be an indirect embryogenesis because they are not predetermined for the development of embryos. Any other somatic cells or organs has to be induced for embryogenic process, and that is an indirect embryogenesis. Here you can see how the somatic embryos are formed under in vitro conditions. So the last and another important concept in tissue culture is acclimatization. So this is a very important concept because even the people have to acclimatize to a certain environment. When they are migrated, when they are shifted from one place to another place, they are subjected for stress of that particular place. In the same way, the plants are also subjected for different stresses when they are transferred from one place to another place. Here we are talking of the transfer of the plant from the in vitro conditions to the in vivo conditions, where the environment is totally different and the plant has to acclimatize itself for its survival, otherwise it will die. So therefore hardening is generally used as the term for acclimatization. So in this technique, the plants are transferred from in vitro conditions to the in vivo conditions, and the plants are generally delicate when they are transferred for hardening. So, and they lose lot of water, so we need to conserve the water. And there are the thin roots, stem, leaves, and the reduced vascular tissues, the poorly developed chlorophyll. They all become the hurdles or drawbacks for the growth of the plant. So in vitro grown plants are adapted to conditions of high humidity, they are in high nutritional supply, they are in low light intensity, and they have a good supply of sucrose. So these conditions will change when they are transferred to the outside conditions. So there will be change in the humidity, there will be increase in the light intensity, and there is a water loss faced by these plants. Somehow they are hardened and they are made into a complete plant and they are shifted. So this is how hardening is done, and you can see how these are transferred to the pots with the peat moss as the medium, and when once they are okay. So this is the last slide to show the hardening process. So I thank you one and all for this presentation. So thank you again, once again, for all my students. Thank you.